Okay, we are now going to start our treatment of genetics. And let's preface this by pointing out that genetics really is the oldest branch of biology. And that is because humans, Homo sapiens, has been practicing genetics in the form of domesticating animals and plants by selective breeding for over 10,000 years. And this ranges from horses in very early times in native Americas, for example, and Europe and the steppes of Europe, to Egyptian cultivation of plants, date palms, for example, and also obviously dairy cattle. The pharaohs of Egypt also were domesticating donkeys, and shown here are the burial of domesticated donkeys. In the Americas, the sunflower was an early cultivar, cultivated in the western United States and Mexico, apparently. Corn is one of the best examples. Corn is derived from a very small plant that grows in the Mexican highlands called teocinte. And modern maize, our modern corn varieties, are all derived ultimately from the domestication of this plant. And it's not that the original farmers who took teocinte had in mind that they would get ears of corn like this, but they selected for features of this plant that did yield increasingly large kernels of corn for eating. And this is what we ended up with after many hundreds and a thousand years of cultivation. And then there's the classic example of peas that Mendel used to discover the laws of heredity. So we will be studying Mendel's experiments with peas shortly. And I only show this to again emphasize that genetics really is the oldest branch of biology. And what is genetics? Here's where our treatment of genetics is going over the next little while, over the next few weeks. Genetics really is information flow. That's what the science of genetics is. There is transmission genetics, which describes the properties of information flow between generations, the way that genes are inherited and passed on from parents to offspring through gametes or through cell division if it's a bacterium. And molecular genetics describes information flow within cells and organisms. That is, this is the central dogma of what we would say molecular biology is. The central dogma of molecular genetics is that information stored in DNA, copied into RNA, is translated into protein. And naturally, within this paradigm, DNA replicates as part of this. So we have DNA replication, transcription of DNA into RNA, and translation of RNA into protein. And so we have two real branches of genetics that we'll be treating over the next few weeks. We're going to start out by talking about transmission genetics, information flow between generations. So let's look at what the types of experiments that Mendel, the discoverer of the modern laws of heredity, were. Let's look at the techniques he was using. And people have been using these techniques for very long periods of time. We have male parts of pea flowers and female parts. And what can be done is the removal by cutting of the male parts so that self-fertilization cannot occur. You can prevent self-fertilization. And in that case, you can grab pollen using a fine brush or human hair even. You can obtain from another flower the male gametes, the pollen, and you can place those on the female parts, the stigma, and the pollen grains will migrate down a pollen tube and fertilize eggs in the ovary of the recipient. And those fertilized embryos will develop into peas, in the case of the pea plant. And those peas can be planted to give rise to plants, which in this case have purple flowers. So you can do these types of crosses then at will. Well, why did it take so long? If we had been practicing practical genetics, applied genetics for thousands of years, why did it take so long for the discovery of the basic laws that govern transmission of information from one generation to the next? And that's partly because the intent of applied geneticists, that is, domestication breeders was to develop better agricultural plants and animals. And their goals really weren't to understand the basic biology of the process. So let's look at an experiment that was reported by an investigator, Gauss, in 1824, well before Mendel did his work. And let's look at his data, and I'm kind of paraphrasing the approach that, I'm paraphrasing the data from his paper, and this is how he reported them. So he grew a plant from a green pea, a green seed, and he grew a plant from a yellow pea, a yellow pea. And he crossed those two plants that grew up from these seeds. And he noticed that all the seeds from that cross, all the peas in other words, were yellow. So he took those yellow peas and he grew them, and he allowed them to self-fertilize. He did not cut off any of the male parts. He allowed self-fertilization to proceed. And this is the results that he got in terms of what his peas looked like after self-fertilization of these individuals. He had some pods had all yellow seeds. There were many pods with both yellow and green seeds. They were mixtures. And there were some pods with all green seeds. So he took green peas from these pods, these mixed pods, and green peas from these all green pods, and he grew those plants up and allowed those to self-fertilize. And he noticed that in that case, all the progeny plants had green peas only, exclusively green peas. Whereas if he took pods with all yellow seeds and he took those yellow peas, grew those into plants and allowed them to self-fertilize, he obtained a mixture again, some pods with all seeds yellow, some with green and yellow seeds mixed. Well, what do we see in this? Gauss was interested in pea breeding, but not necessarily interested in the basic laws of heredity. Or if he was, he certainly was missing a key ingredient. And what do we know? What do we see, rather? What do we observe about this type of data? What is missing in this description of data? Let you think about that for a moment, and it'll probably come to you, is that there is a lack of quantitative data. There are no numbers associated. Some, many, some. There's no quantitative data. Whereas Mendel, an Austrian monk who clearly had a clerical bent and was an excellent experimentalist and data recorder, Mendel, let's look at his data, which was basically derived, his initial experiment was initially derived from exactly the same type of cross. So let's look at the way that Mendel recorded his data. And Mendel published in 1866, much later than Gauss did. So here we have, the first thing that Mendel did is he was after the laws of heredity, and he established true breeding lines of plants. That is, plants that only produced yellow peas, a line of plants that only produced yellow peas, and a line of plants that only produced green peas. And having these true breeding lines then allowed him to breed with confidence and would allow him to determine with confidence the results of his experiments in terms of the properties of inheritance. So he calls the first generation the first filial generation, or the F1. So all seeds are yellow, just like Gauss. When he crossed a pea plant that was grown from green pea and a pea plant that was grown from a yellow pea, all the peas resulted were yellow. And he grew those into plants, and he allowed those to self-fertilize, just like Gauss had done. And then here's where Mendel gets quantitative. He counted the number of green and yellow peas here. And out of 8,000 total seeds in one experiment, approximately 6,000 were yellow and approximately 2,000 were green. That is, there was a 3-to-1 ratio of yellow to green. 3-to-1 of yellow to green peas in this F2 generation, So what did he do? He took some of these green peas and grew those into plants, and he allowed those to self-fertilize. And just like Gauss, when that was the case, he got all green peas produced. In other words, he had established a new true breeding line of plants grown from green peas and allowed to self-fertilize. Whereas if he took some of these yellow peas, he took 519. Mendel grew a lot of plants and looked at a lot of data. So he took 519 of these yellow peas, grew those into plants, allowed them to self-fertilize. And of those 519 plants, 166 gave all yellow seeds. That is, they bred true for the yellow pea appearance. But 353 did not. They did not breed true. That is to say, when those 353 plants were allowed to self-fertilize, they gave rise to progeny peas that were mixed yellow and green. So there, in this case, then, what we have is if we compare these, what we have is a 1/3 true breeding and 2/3 of the plants grown from yellow seeds were not true breeding. We'll come back to these 1/3 and 2/3 shortly, but keep that in mind for now. So let's see what Mendel did with this data. But before we do that, let's just point out that, in fact, he could do this with other traits. He could cross, now not looking at pea color, but rather flower color, he would cross true breeding purple flowered plant from a true breeding purple line and a white flowered plant from a true breeding white line, and he got all purple offspring in the F1 generation. And when those were self-crossed, self-fertilized, he got a 3-to-1 ratio of purple flowered plants to white flowered plants in the F2 generation. Let's not worry about the F3 generation right now. And let's look at what Mendel's model was that he used, that he derived from his data. And here's what his model involved. He said that for every trait that you're looking at, for example, yellow peas versus green peas, that there are two factors, two elements. In the case of the true breeding yellow, he designated those as two capital A symbols representing those two elements, those two factors. Whereas he used little a's to designate the two factors in the true breeding green plants. And Mendel said he could explain his data if there are two factors that govern a particular trait, like the pea color, that when plants produce reproductive cells, egg cells or pollen cells, or in animals, egg cells and sperm cells, those gametes, those reproductive cells, those sex cells, only obtain one of the two factors that are present in a given individual. So that a little a plant would only produce pollen cells that contain the little a factor, which would specify green peas. Whereas egg cells from a true breeding plant would only obtain one of the big A factors that would specify yellow. And so that when these combined by fertilization, that is when he did his parental cross, he obtained an F1 generation that had one large A factor and one small A factor. And as you know, all these F1 peas were indeed yellow. They were not green. And we'll talk about dominance and recessiveness in a moment. But right now we have a mixture, an F1 generation that has a large A factor and a small A factor. And when those yellow seeds were grown into plants and self-fertilized, we can ask what kind of pollen and eggs they would produce. Well, remember, Mendel was saying that he can make his data work if when sex cells are produced, only one of the factors that are present in an individual finds its way into a reproductive gametic cell. So here, if we have individuals that are a combination of these factors, big A and little a, they will produce, for example, pollen that half of which contains a big A and half of which contains a little a. and they will produce eggs, half of which contain a big A factor and half which contain a little a factor. And then if we allow the random mixing of these pollens with these eggs, we would get the following. 1/4 of our progeny, 1/4 of the progeny would contain 2 big A's. They would result from the fertilization of a big A containing egg with a big A containing pollen. 1/2, if we look at these two together, 1/2 of the progeny peas would contain 1 little a factor and 1 big A factor. And that is, they would result from the fertilization of a big A egg by a little a pollen, or alternatively by a little a egg by a big A pollen. But they would be equivalent in terms of the factors that they inherited. They would be a mixture just like their parents would have been. And then 1/4 of the progeny would inherit a little a from a little a pollen and a little a from a little a egg, and they would have 2 little a's. So this constitution of factors is just like the true breeding green grandparent up here. And this combination, the 2 big A's, is just like the grandparent that from the true breeding yellow line. Whereas these mixture F2 peas would be just like their F1 parents. So what does this result in? This results in a 3-to-1 ratio of yellow to green. So we have yellow and green in a 3-to-1 ratio. That is 3 yellow to 1 green. And we can immediately see that of the yellow peas in the F2 generation, only 1/3 of them will be true breeding, in the sense that if they are grown into plants and self-fertilize, they will give rise to only yellow peas, as their grandparents were true breeding big A, big A. Whereas of the yellow peas in the F2 generation, of the yellow ones, 2/3, when self-fertilized, will give a mixture of green to yellow peas in the same ratio, exactly the same ratio, 3-to-1, yellow to green. So these ratios spoke to Mendel in a profound way, and excuse me, and allowed him to develop his first law. And here is his first law. Each trait, whatever it is you're talking about, curly hair, straight hair, brown eyes, blue eyes, purple flowers, white flowers, green pea, yellow pea, every trait is governed by 2 particles, and one is inherited from each parent. And the 2 particles do not influence each other in any way in an offspring. But when that offspring grows up and becomes sexually mature, those particles separate uncontaminated. They do not influence each other, in other words, into gametes when reproductive cells form. And an unstated corollary is that for plants, for peas, any pollen cell can fertilize any egg cell. There's random fertilization. Consider if that weren't the case. If there weren't random fertilization, let's say that big A pollen could only fertilize big A eggs, and little a pollen could only fertilize little a eggs, well then you would not get a 3-to-1 ratio of the following combinations. You would get little a, and big A in the F2. That is, you'd get half yellow and half green. But we have a 3-to-1 yellow to green ratio from a monohybrid cross. We call this a monohybrid cross because we're looking at one character trait, and we have produced hybrids in the F1 because we've crossed 2 plants or 2 individuals that differ in that trait. So we've hybridized, and it's a monohybrid cross because it's we're only looking at one character trait at this point. So now we can introduce modern terms. We can take Mendel's law and put it into modern terms. So let's talk about his particles. The particles are known as alleles. So for every trait then, there are two alleles that govern that trait. And as we've seen, those alleles can be dominant or recessive. In the case of the P's here, the big A allele, the big A version of the gene, in other words, is dominant over the recessive little a, because in this combination, we have the appearance of the big A character, which is yellow seeds in this case. And we call the little a here, which specifies green peas, we call that a recessive allele because it recesses for a generation in a monohybrid cross. It's present in the parental generation here. It disappears in the F1, and then it appears again in the F2 when the F1 is self-fertilized. So the green pea trait, which is specified by the small a allele, recesses for a generation. So we call that the recessive allele, and the one that dominates over that, we call that the dominant allele. And now we can introduce the terms phenotype and genotype. And phenotype and genotype genotype, let's take genotype first. Genotype can refer to two things. One is that you can have the genotype of an individual. So an individual, let's say, could be big A, and or it could be little a, and or it could be big A, little a. So all of these are genotypes. And we refer to these two genotypes as homozygous genotypes because both alleles are the same in these homozygous genotypes. Whereas in this genotype, we say that we have a heterozygous situation because there are two different alleles present. Now, genotypes govern phenotypes. So phenotypes, phenotypes we can define as the appearance of a trait in a given individual. It is the genotypes that determine the phenotypes. So in our previous cases, the homozygous big A genotype and the heterozygous genotype gave a yellow phenotype, whereas the homozygous small a genotype gave rise to the green phenotype. So genotypes determine phenotypes, and we have heterozygous genotypes or homozygous genotypes, but we shouldn't forget that there's also the genotypes of the gametes. So we could say this pollen grain, this pollen cell has a little a genotype, and this one has a long. Gametes have genotypes as well. So here we have a little a genotype for this pollen cell, a big A genotype for that pollen cell, a big A genotype for that egg cell, and a little a genotype for that egg cell. So gametes have genotypes as well. Now, Mendel went on to test his law. He wasn't satisfied with just coming up with an explanation or a law that would explain his 3-to-1 ratio of recessive phenotypes to dominant phenotypes in the offspring of a monohybrid self-fertilization. He tested the law, for example, by crossing, let's say, heterozygotes by homozygous recessives. So, for example, he would cross big A by big A, as he originally did, little a, so these are, let's say, are yellow, and these are green. And he would get these F1 heterozygotes, which were yellow. These were yellow. And he would cross those, he would grow those up into plants and cross them by green plants, like these. And he asked, or he predicted what ratios of phenotypes he would get in the offspring of this cross. And think about that for a moment. Is there anything magical about that 3-to-1 ratio that Mendel came up with? No, there isn't. And Mendel used his understanding, his law, to predict accurately what would happen here. Well, let's look at it. Let's draw it out. So if we use our little Punnett square method, so-called Punnett square after a geneticist named Punnett, to see what kind of gametes, genotypically speaking, this individual would produce, it would only produce little a gametes. And we don't even need to make a second column because 100% of the gametes will contain little a. There's just no other alleles present to find their way into gametes. Whereas these heterozygous individuals will produce half of their gametes with a big A genotype and half of their gametes with a little a genotype. And that then should produce 1/2 of the offspring of this cross should be heterozygous big A little a, and the other half of the offspring should be homozygous little a. So we should get 1/2 yellow that have a big A little a heterozygous genotype and 1/2 green that have a little a homozygous genotype. And that is exactly what he got. So Mendel actually became the first person in history to be able to accurately predict the ratios of phenotypes of offspring from a given genetic cross, despite the thousands of years of domestication in which people were interested in being able to predict or determine what percentage of phenotypes would be available in a particular cross. Mendel was the first one to discover a law of heredity that would allow the accurate prediction of this. And he tested other traits. In addition to doing the experiment that we just talked about, in which he made specific predictions based on his law and had those predictions confirmed, he tested other traits. In fact, he looked at 7 traits. He looked at flower color, purple and white, and yellow and green peas. We've already talked about that. Round and wrinkled peas, green and yellow pea pods, inflated pea pods versus constricted or wrinkled pea pods, axial flowers that would grow from the branches on the side branches of the plant versus terminal flowers which would be growing from the end of the plant, the top of the plant. Plant height as well, tall versus short. And in all 7 of these cases, he found when he made monohybrid crosses, he would get a 3-to-1 ratio, roughly, of the dominant phenotype to the recessive phenotype in the self-crosses of the F1 hybrids. And there was always a dominant allele that he could assign to the dominant phenotype. So for purple, he would have, let's say, big P is the purple flower, little p is the white flower. The F1 hybrids would be big P, little p heterozygotes. And when those were selfed, he would get a 3-to-1 ratio of purple to white. And we would be able to predict what the genotypes of those would be. And we'll run through some examples of that in a little while. So he tested this with other traits and therefore became convinced of the universality of his first law. But he didn't stop there. In addition to testing all these traits that we've just mentioned, and here are the F2 ratios, he tested what would happen if 2 character traits are followed simultaneously. That is, instead of doing monohybrid crosses, he looked at the phenotypic ratios produced by the self-fertilization of dihybrid, 2 hybrid crosses. So now what Mendel did is to follow the inheritance of 2 traits simultaneously. So here we have a round yellow plant where round is dominant to wrinkled peas, big R, and yellow peas we already know are dominant to green peas. So we specify yellow with big Y, green with little y, wrinkled peas with little r, and round peas with big R. So here we have a homozygous big R, big Y genotype. And we have a true breeding line of plants like that. Mendel was careful to establish true breeding lines. And here we have a little y, little y, double homozygous genotype that produces wrinkled and green peas in a true breeding line of plants. So Mendel crossed these and observed in the F1 generation, not surprisingly, given his previous results, that the dominant alleles were dominant and the recessive alleles recessed. So in this double heterozygote now, big R, little r, big Y, little y, in the double heterozygote, we had a double dominant phenotype. That is, round peas and yellow peas. Now, what would happen then if we self-fertilize these? And this is what Mendel got. These are his ratios that he observed in the F2 generation. And let's see why that is true. Well, we just have to ask ourselves what type of gametes these F1 individuals would produce. And so we can say, well, in one type of gamete will inherit a big R allele, and that gamete could inherit also a big Y allele, remembering that only one allele from each gene that governs a particular character trait, only one allele finds its way into any given reproductive cell, any given gamete. So we could have big R going into a gamete with big Y. We could have big R going into a gamete with little y here. We could have little r into a gamete with big Y and little r with little y. That is, there are 4 genotypes of gametes made possible by this genotype. And so if we self-fertilize, then we will have sperm with those 4 same genotypes as these eggs. And if we look at random fertilization, we will get the following genotypes. And there are 16 possible combinations. Note that only 1/16 here, 1/16 will have a wrinkled green phenotype. That is, that is the double recessive phenotype. And that is because if only 1/4 of the sperm are little r, little y, and only 1/4 of the eggs are little r, little y, then it would be 1/4, the probability of getting that type of fertilization event is 1/4 of the eggs times 1/4 of the sperm equals 1/16. And indeed, Mendel saw that 1/16 of the progeny in these dihybrid crosses had the double recessive phenotype. Well, many more have the double dominant phenotype. So here we have round and yellow are the double dominant phenotypes. 9/16. And you can add these up and see why that is true. Of those 9/16, though, only 1/9 are, if grown into plants and self-fertilized, will be absolutely true breeding. That is, 1/16 are homozygous for both alleles, for both recessive alleles. Likewise, only 1/16 are double homozygotes. They're doubly homozygous for the dominant alleles in this case. Here they were doubly homozygous for the recessive alleles. Here they are doubly homozygous for the dominant alleles. All the other that are the double dominant phenotypes are heterozygous for at least one of the 2 genes that we're talking about. So all of these other all the other 8/16 that have double dominant phenotype have at least one, are heterozygous for at least one of the 2 genes that we're looking at. Oops, that one's not. All right, so there's another one, there's another one, and there's another one. All right, now, what about the other 6/16? Well, of those, we have 3/16 that are dominant have the dominant phenotype for one trait and recessive for the other. That is, 3/16 are round, which is a dominant phenotype, and green, which is the recessive. And other 3/16 are recessive for the first trait and dominant for the second trait. That is, these are reciprocals of each other. And we can see why that's true. For the round green peas, we have this genotype, which is homozygous for big R and homozygous for little y. Here's another one of those 3/16, heterozygous for big R, homozygous for little y. And here's yet another one, heterozygous for R and homozygous little y. Of course, because of these 3/16, we're talking about ones that have the recessive phenotype for one of the two character traits, of course those have to be homozygous recessive in order to have that recessive phenotype. And likewise, you can go through this on your own for the wrinkled yellow seeds. That would be here. You're homozygous for the little r allele, that is, you have a wrinkled phenotype. And you are either homozygous big Y, yellow, or heterozygous big Y, little y, and you will also have a yellow phenotype in that regard. So Mendel could explain his 9:3:3:1 ratio. We commonly draw this as 9:3:3:1 ratio. This is a result of a dihybrid self-cross. It's the characteristic ratio of a dihybrid self-cross. Hybrid for two traits, and we self-fertilized. He could explain his 9:3:3:1 ratio by stating his second law, and that is as follows. It's the law of independent assortment, and that it reads as follows. During the formation of gametes, then, the segregation of alleles at one locus, and for locus, let's just write in gene. Gene is the same thing as a locus. The segregation of alleles at one gene is independent of that of the segregation of alleles at any other. What does that mean? Well, what that means is that in the formation of gametes from this heterozygote, let's say, there's an equal probability that if a gamete receives a big R and a big Y, that a gamete will receive a big R and a little y. That is, the Y alleles, which govern color, and the texture alleles, which govern round or wrinkled, the alleles for those traits segregate into gametes completely independently of each other. Let's imagine that weren't the case. If that weren't the case, what if when a genotype, a double heterozygous genotype like this, that is a dihybrid heterozygote, if let's say when this formed gametes, if a gamete received an R, it always received a Y, a big Y. Big R always went with big Y for some reason. And likewise, let's imagine little r always went with little y. Well, then there would only be two types of gametes formed. And if you look at sperm and eggs, you would have big R, big Y, and little r, little y. And then you would end up with a 3:1 ratio of double dominant phenotypes to recessive phenotypes. So you would have big R, big Y here, big R, little big Y, heterozygote here, big R, big Y, little y. Here, that's a little y there. So here we would get a 3:1 ratio of dominant, double dominant, to recessive, double recessive for both traits. Here we would have we would have wrinkled green, and all the other three here would be round yellow. So if this were the case, if there weren't independent assortment of the alleles into gametes, such that in the case drawn, you could get 4 possible genotypes of gametes. If that weren't the case, you would get something other than a 9:3:3:1 ratio. Yet that is what Mendel got repeatedly when he repeated these experiments for different traits monitored in dihybrid crosses, and therefore his second law seemed like a universally applicable law. And we now know the basis for this, and as we see when we study, when we draw together the behavior of chromosomes with Mendel's alleles, we will see that to be true. And that's what we'll cover in the next part of this lecture.